So in both groups, we were able to begin the introduction to the cancer lecture. And we just barely got started talking about the fact that cancer is a genetic disorder of DNA mutations. And when those DNA mutations affect the function of genes that are involved in regulating fundamental cellular processes, these are the types of mutations that have the ability to, in some circumstances, lead to cancer. So let's go through what types of genes we're talking about when we say key genes that regulate fundamental cellular processes. So in your notes, these are referred to as the principal targets of genetic damage that lead to cancer. So the first is going to be proto-oncogenes. Now, for each of these are normal genes in our cells that get mutated. So a proto-oncogene is a gene that's in your cell, and it normally has, a good job to do. It is a gene that signals that the cell should divide. So it says in your notes, it's the go signal for cellular division. So proto-oncogene is a key gene to regulate cell growth and differentiation. It tells it when it's time to divide. And that's a really important signal for a cell to get to know when to do that. When a gene that's a proto-oncogene gets mutated, we start calling it an oncogene. And we call it an oncogene because it's a gene that can transform a normal cell into a cancerous cell. So a mutation in a proto-oncogene, a normal gene that says go to cellular division, a mutation that happens is that this gene is either overexpressed, meaning there's too much of it, right? There's an additional amounts of this gene available, or it's constitutively active. So on our DNA, the genes are there, but they can be in an inactive state where you couldn't get the RNA off of it. Constitutively active means it's always on. You're always expressing that gene. So if a gene that tells a cell to divide is either overexpressed or always active, it's telling that cell to continue dividing forever. It never stops telling the cell to divide. So there are a host of carcinogens that can cause a proto-oncogene to mutate into an oncogene. Once the oncogene is there, meaning it's either an overexpressed or constitutively active proto-oncogene, it will force the cell to grow and divide, even in the absence of normal cellular cues. Normal things that we have to tell the cell to divide, we don't even need them anymore. It's just go. This is unregulated cell growth. A continually on grow and divide signal is now a cancer cell because it will continue to divide and grow, and there's nothing that will stop it. So that would be the activation of a oncogene. Another gene that is frequently involved in cancer development is tumor suppressor genes. These are genes that encode proteins that regulate the cell cycle. And as the name implies, tumor suppressor, these are stop signals. So where a proto-oncogene was a go signal, tumor suppressor genes are a stop signal. They tell the cell not to divide. These are also the genes that can be involved in sensing and detecting if genomic damage takes place. So I think I mentioned this in the second lecture, and maybe not the first, but mutations to DNA are normal. I mean, when you copy the DNA over, it's normal to make a mistake here or there. And that's not life-threatening necessarily. A lot of them are very small changes. And you can have caretaker genes that are checking to make sure everything is good. So tumor suppressor genes can fall in this category where they're looking at the genome to determine if everything is correct, and if something is wrong, trying to figure out what to do about it. Either, fix it, correct it. It might be a reason that the cell has to go through apoptosis because the damage is too significant. They're an important caretaker gene, excuse me. So if a mutation occurs to a tumor suppressor gene, and that mutation ends up inactivating the tumor suppressor gene, the mutation turns the stop gene off. So if this tumor suppressor gene is normally kind of having their brakes on any signal for cell division, the mutation turns that gene off, takes the foot off the brakes, and now there's no longer a signal telling the cell not to divide. So inactivation of a tumor suppressor gene results in carcinogenesis or cancer growth because you've turned off the stop signal for cell division. Another family of genes are genes that regulate apoptosis. So apoptosis, again, is the programmed, planned cell death. And there are genes that are anti-apoptotic genes. These are genes that prevent apoptosis from happening. And when we have a healthy, regulated cell, these can be important because you don't want healthy cells undergoing apoptosis. In cancer, you can get mutations of anti-apoptotic genes that cause them to be overexpressed. If you have too many anti-apoptotic genes, then you can't trigger apoptosis when you need to, and it can lead to uncontrolled growth. There can be pro-apoptotic genes that trigger apoptosis, and a mutation that causes a loss of function of a pro-apoptotic gene would prevent that cell from getting the apoptosis signal, and again can trigger malignant growth. The next category of genes are genes that are actually involved in the DNA repair. And again, these kind of fall into that caretaker category. When you have damage that has been detected, there are genes that can help fix it. If the genes that help fix DNA damage become mutated or inactive in some way, then you can't fix DNA mistakes. And that leaves the cell vulnerable to the development of cancer. Just one example, because it's probably one of the most well-known, and then Angelina Jolie is a celebrity that made this story even more kind of in the media, is the BRCA genes. So the BRCA genes stand for breast cancer gene, and they produce proteins that normally go and repair DNA. So when there's double-stranded breaks, they can go and repair. If the BRCA gene is not functional, it will contribute to the development of cancer. So this is a type of breast cancer that we do know the genetic cause of. So when you've had a family member that's had cancer, a BRCA positive cancer, first of all, there's different treatments for that. That's kind of beyond the scope of this lecture, but family members, like offspring, can get tested to see if they have that same gene mutation. So Angelina Jolie very kind of famously, publicly shared the loss of her mother at a young age, and she had a BRCA positive cancer. And so she was tested and was found positive, and she underwent a radical mastectomy and I just said that backwards. She went through a hysterectomy and double mastectomy, and was very public about that journey to try to support other women to do preventative actions when they have a genetic possibility of developing a certain type of cancer. So if you've heard that story in the news, that's where this gene kind of ties in. So those were examples of specific genes that get targeted and can end up causing neoplasia, which is new uncontrolled growth of cells that is not under physiological control. When this growth comes together, we often refer to it as a tumor, And again, this can be malignant or benign. We already talked about cell growth in this unit, right? We talked about 2 adaptive changes to a cell that would fall into the category of growth. Cells can get bigger with cellular hypertrophy. They can increase in number with hyperplasia. And these are adaptive responses that very notably are reversible. They can go back and forth depending on the stimulus. When we have uncontrolled cell growth, the formation of neoplasms is non-reversible. So regulated cell growth is a reversible process. Unregulated cell growth is not reversible. That's a big, big difference right off the bat. We describe the tumor or the neoplasm based on where it is in the body and what cells originally kind of developed the tumor. So a primary tumor is a tumor that arises from cells that are normally local to the given structure. So they carry some origin of the cell type in which they're growing. Whereas a secondary tumor arises from cells that have traveled from another part of the body. So it's like a hepatocyte that ends up growing in a bone. And it's not a bone cell, it's a hepatocyte, but it's a tumor that's growing with hepatocyte-like cells. That would be a secondary tumor. So we're going to describe cancer a lot as being this abnormal cell growth. So it's important to kind of step back and say, well, what's normal? If you're going to biopsy something and look at it under a microscope and define it as being abnormal, let's first just talk about what's normal. So cellular characteristics of normal tissue include cells that are uniform in size and shape. So if you look into this slide, you can see the cells within it all basically have the same size. They all basically have the same shape. They look alike. This picture is just normal, healthy liver parenchyma. They are all at similar maturity. Like if you're looking at kind of the nucleus of these cells, we don't see a lot of cells undergoing cellular division. These are adult cells. They're fully grown up. They're not actively dividing. If you look at the nucleus, the nuclear structure looks normal. All the nuclei look the same, and there's not abnormal shapes in them or very dark or, something like that. So this would be kind of characteristics of normal tissue. To talk about normal, we have to make sure we define the term differentiation. So obviously this is written out in your notes, but it's the process by which normal cells undergo physical and structural changes as they develop to form the tissues of the body. What that means is every cell begins as a pluripotent stem cell. It could be anything it wants to be. It's just sort of an empty blob. Like, tell me what you want me to do and I'll be it. So a pluripotent stem cell can turn into basically any other cell in the body. It just has to get the right signals to divide and develop its adult structure. So it needs to undergo physical and structural changes to become a specific cell of a specific tissue. So I gave you the examples that we have stem cells in our epidermis that have the ability to differentiate into skin cells. Once a cell is an adult, it fully represents the cellular structure and function of that type of cell. So it gives it its specific physiological functions. So when we talked about the cellular characteristics of normal cells, they're uniform in size and shape. They look the same and they function the same because they are fully differentiated cells. Fully differentiated is synonymous with like all grown up adult, you know who you are, you know what you're going to do, and you're not going to change. Fully differentiated. So if we're comparing normal tissue to malignant cells, and on the right we have a slide of malignant cells. And if you look at this slide, you can see that the cells are kind of abnormally sized. There's some big cells, there's some small cells. And the dark purple is all the nuclei. And there's some really weird looking nuclei. The arrows are kind of pointing to these horseshoe shaped nuclei, kind of from a not specific thing. Things look kind of funny on the slide. The reason the cells alter their visible appearance is because with malignant cells, this process of being fully differentiated gets lost. And in malignant cells, that differentiation can either be altered or can be completely absent, meaning the cell doesn't even really resemble the original parent cell that it started from. So if we walk on the journey of how a cell gets there, the term dysplasia is sort of a general category indicating a disorganization of cells in which an adult cell varies from its normal size, shape, and organization. So the size, for example, you can get abnormally big cells. That's a really common finding where they're just bigger than they should be. You can see changes in the nuclei of the cell. You can get what's called hyperchromatic nuclei where they get real dark. That's kind of represented here by those dark hyperchromatic nuclei. The lack of organization can be seen in what's called mitotic figures, evidence of cells undergoing mitosis. When they vary in size and shape, kind of the fancy word for this is pleomorphism. Pleomorphism means a variation in size and shape. When you see on a slide cells that are abnormally large, changes in the visibility of their nuclei, how dark they are, when you view pleomorphism on the slide, this marks a tissue for being at risk for developing an invasive cancer. And if we look at this cartoon on the bottom of the slide, it kind of takes us on the journey from normal cell to this category of dysplasia. And the first step is metaplasia. Metaplasia is the same metaplasia we talked about in like the first lecture in this unit. This is a cellular adaptation to a particular stress. And it's a reversible adaptation, right? It falls in the same kind of general category as hypertrophy and hyperplasia and atrophy. Metaplasia is when a well-differentiated cell is replaced by another well-differentiated cell. It's not the cell that's supposed to be there, but it's still a well-differentiated cell. And I kind of mentioned this when we talked about metaplasia. The kind of quote-unquote problem with metaplasia is obviously that you lost whatever the function of the original cells were, and they were probably there for a reason. But the other big problem is that metaplasia is kind of the first step towards dysplasia. Metaplasia is still an adaptive and reversible process, but it has the tendency to lead into dysplasia. And so I just gave another example in your notes of this, where in the respiratory tract, you can get epithelial metaplasia, where columnar epithelium, which is ciliated with goblet cells scattered among to help protect our airways, can be replaced with squamous epithelium to prevent the destruction that the irritant is causing. So what the cell looks like gives us a lot of information about what's going on in terms of the development of cancer. So when individual is investigating whether or not a patient has cancer, they can do a biopsy and look at the cells under a microscope. And they basically ask the question, how well does the tumor resemble the original cells? So if it closely resembles it, which is kind of like here, if you look at this, it's uncontrolled cell growth, but man, they really look pretty similar. We would call that a well-differentiated tumor. They still carry the structure and function of the original cell. They're just growing outside of physiological control. If they kind of sort of resemble it, They have some of the structure, maybe some of the function, but they're starting to look kind of funny. We call that moderately differentiated. And when the cells do not resemble the parent cells, that is poorly differentiated. When a cell completely loses its structure and function, we call that anaplasia. Anaplasia is a state of complete undifferentiation. And where well-differentiated cells in a tumor are almost always, not almost always, it is a safe association to say this is a benign tumor. Anaplastic cells are a reliable indicator of malignancy. This differentiation scale, how we describe the cells, can actually be used to grade the tumor. So you can see that as the number gets higher, we're getting closer and closer to anaplasia, marking the tumor as more likely to be a malignant neoplasm. So let's touch on this malignant versus benign question, because I've referenced that neoplasms can be both. We've kind of hinted a little bit, starting to hint at how the benign is different from the malignant. But I first want to just drive home one important fact about tumor growth is that it doesn't have to be malignant to cause problems. So a malignant tumor has the capacity to spread to other areas of the body, and it is worse, but any uncontrolled growth comes with issues. So it can cause problems even if it's not cancer. That term benign can sometimes mask the problems related to a benign tumor. So benign means not cancerous. It doesn't necessarily mean that it's good, no problems. Okay. So this list in your notes is problems that you could have with both malignant and or benign tumors. Okay. So it doesn't matter if it's malignant or not. These are problems that can happen when a tumor is growing. One, you can get impingement on adjacent structures. So a brain tumor can be benign, but it could still kill a patient from increased pressure on the brain. Okay, so if it impinges an adjacent structure and impedes that function, that can have a huge problem. Another problem that can arise is that the tumor can have functional activity. What that means is if a cell is dividing outside of regulation and that cell still has function, it's probably going to be doing its job super, super good. So we see this most common when we have endocrine tumors, where there's endocrine cells that can make a hormone. You start making a tumor, and that tumor starts making tons of the hormone. That can overwhelm the system and cause a huge physiological response from excessive hormone production. In fact, in terms of this issue with hormones, benign tumors can almost be worse for this because they often maintain the function of the parent cell, where malignant can become anaplastic and really has no related function to the original adult. So like a big example of this are thyroid tumors that get very large and create a lot of thyroid hormones and give patients the symptoms of excessive thyroid hormone. Any tumor that's growing without regulation can ulcerate through other areas. They can cause bleeding and cause infections. This is a big problem. For a tumor to be considered benign, this should kind of fit with what we've talked about so far. Because they are typically a more well-differentiated cell in the uncontrolled growth, they are typically the cells are typically fairly uniform in size. Their nucleus is pretty regular in size. They have a low nucleus to cytoplasm ratio. So a typical nucleus to cytoplasm ratio is somewhere between 1:4 to 1:6. There should be a lot more cytoplasm than nucleus. And the nuclei do not stain dark, so they're not those big dark hypochromatic markings on the inside. These would all be helpful in the identification of cells that are growing uncontrolled but will not spread to other areas as cancer would. So we use different language to name a benign tumor compared to a malignant tumor. If just for a minute you turn to your next page in your notes, I have this all as one chart where I have the benign and malignant together. So if you'd like to look at them as I'm talking, that's where this chart is. I didn't want you to think you didn't have it. So the tumor is named by the tissue of origin. And if it is a benign tumor, we put an oma at the end. That helps us identify immediately by the name that it's a benign tumor, a lipoma, a osteoma, for example. Okay, so whereas we went through the cell types in normal tissue and how benign tumors may look, a malignant neoplasm's cellular characteristics are that the cells are extremely variable in size and shape. Again, this is pleomorphism. And when you look at the nucleus of the cells, they vary in size and shape. So when you're looking at the dark staining of the nucleus, they look different between them. That is referred to as nuclear pleomorphism. In malignant cells, we can see an increased nucleus to cytoplasm ratio. In fact, it can get as high as 1-to-1, where the nucleus almost seems to take up all this, almost all the space where the cytoplasm used to be. The nuclei stain very dark, and this is largely related to the high DNA content within the malignant cells. You can start to see the appearance of the nucleolus. This is like a dark dot in the nucleus. The nucleolus is where ribosomal RNA is formed. So the visual appearance of the nucleolus is really telling you this is a visible sign of increased protein synthesis in the cell. You also start to see those mitotic figures or signs of rapid mitosis. And you can see one in this slide right in the middle. Can you see that guy with the nuclear material separating out into the two sides of the cell, clearly undergoing cellular division in this slide? So we mentioned that we put oma at the end when it's a benign tumor, but all tumors, including those that are cancer, are named by origin. So we're going to go through the three main cell types where cancer develops: epithelial cells, solid mesenchymal tissues, and then cells of the blood and lymphatics. So when your cancer originates from epithelial cells, it is called a carcinoma. This is by far the most common cancer. 80 to 90% of cancers are epithelial in origin, which makes sense because of the type of cell. These epithelial cells cover, the whole outside of our body. It's our skin. And it makes up the tissue that lines the inside of our bodies and every one of our organs. It's a type of cell that divides a lot, so it has the chance of making more mistakes, and that is why it's a very common type of cancer. Solid mesenchymal cancers. Mesenchyma are when loose cells are embedded in a mesh of proteins and fluid, kind of that extracellular matrix. This is the tissue that gives rise to connective tissue. So solid mesenchymal tissues give rise to sarcomas. Sarcomas can fall into two main categories: bone and soft tissue. So bone sarcomas kind of are a little more self-explanatory. When we say soft tissue sarcomas, we're talking about, it says in your notes, supportive tissues. So we're talking about tendons, muscle, fat, blood vessel, nerves. That all falls into the soft tissue sarcomas category. So the naming of the neoplasm, we went through kind of the middle column when we talked about benign neoplasms. For malignant neoplasms, they are again named by the tissue of origin. So if the tissue of origin is fat, the beginning is going to be lipo. If it's benign, it's a lipoma. If it's malignant, It starts with the tissue of origin and it ends with the cellular origin of the cancer. So it would be a sarcoma. So it's telling you that this is mesenchymal tissue and it's fat. Similarly, cartilage, right? We start with the chondro as to tell you the tissue of origin. Chondroma would be benign. Chondrosarcoma tells you it is malignant of the soft tissue that falls under that larger category of all of our sarcomas. So the name itself gives you a lot of information on the type of cancer it is and where it originated. So we talked about the difference between benign and malignant cancer cells way down on the cellular level, which is when they would do a biopsy and look at this under the microscope. But macroscopically, you can also appreciate differences in tumors that indicate benign or malignant. You can see growths in both of these livers on the left and the right. One of these tumors is benign and one of them is malignant. Even on a very gut level, probably one of them jumps out at you as looking maybe quote unquote worse, but I don't want to make you think that just because one is larger, it's worse. The tumor on the left is a hepatic adenoma, so it's a benign tumor. One of the reasons that you might be able to tell that it's benign, and it's not only because it's obviously smaller than the tumor on the right, but it has very clear edges, right? You can almost see the line. I don't want to put my pen right on it because it will cover it, but the line, the edge of the tumor is very well defined where it's separated from the organ. So a well-defined border. It's not encroaching into the tissues. It's kind of contained within its own walls. You can also see it has a kind of a green color. And benign tumors often maintain the function of their cells of origin. So there's cells in the liver that can make bile. So this tumor appears to be currently making bile. So it has some function of a normal hepatocyte, but it's an uncontrolled new growth. As opposed to the tumor on the right, which is a malignant hepatocellular carcinoma, which has less clear borders. You can see it infiltrating into the liver. And then the tumor itself looks less consistent. It has a variable appearance, among the tumor, especially when you compare it to kind of the commonality of the visible tumor on the left. So we've gone through epithelial cells named carcinoma, mesenchymal tissues called sarcomas, and that leads us into when you have cancers involving the cells of the blood and the lymphatics. We really fall into three big categories, and we're going to talk about lymphomas, which start in lymph nodes, leukemias that start in the bone marrow or the blood, and then myelomas, which is cancer of plasma cells. We will say these again, but if you're writing them down as you go, I realized I kind of rattled those off pretty quickly. So lymphomas start in the lymph nodes, leukemias start in the bone marrow or blood, and myelomas are cancer of plasma cells. So to understand the cancer of blood cells, it is important to hold in our mind how our blood cells are formed. So you saw this figure before in our lab values lecture when we talked about the circulating cells, adult circulating cells in our blood. And we mentioned that all of our blood cells start from a stem cell. And the stem cell is first divided into two categories: myeloid stem cells and lymphoid stem cells. We're going to first talk about the type of cancers that originate from the myeloid stem line. Now, in general, myeloid malignancies very rarely go to the lymph node. So if it is a cancer that started in the myeloid stem line, it's almost always going to be a leukemia. It's very, very unusual for a cell that originated from the myeloid stem line to develop into a lymphoma. So we're talking about cells that originate from the myeloid stem line. And you can turn your page just to have the place where we have the notes where we actually talk about myeloid neoplasms. So myeloid neoplasms are cancers that arise from myeloid progenitor cells. So if I go back a minute, we'd call these guys here myeloid progenitor cells. So myeloid neoplasms arise from the myeloid progenitors. And in general, these cancers cause proliferation, that's cellular growth, that involves the bone marrow, and it ends up replacing normal elements. So because of this accelerated growth in the bone marrow, it kind of crowds out the space and the room and kind of the factories for other elements to be developed. So let's first talk about myeloid leukemia. So the name leukemia means that the malignancy is an abnormal growth of leukocytes, thus the leuc in leukemia. The disease of myeloid leukemia causes the cells to stop differentiating. So again, differentiation is the process by which a stem cell develops the structure and function of a fully adult cell. So the cancer tells the cells to halt their differentiation, but then also stimulates rapid cellular proliferation. So you make more and more and more cells, but you don't give them a chance to grow up. So you start making more and more and more baby cells. So because they're baby cells and their differentiation was halted, they don't grow up to have the full function of what that adult cell would have had. So myeloid leukemias are noted by the circulation of immature myeloid blasts. And blast cells, if I just go back a slide, blast cells are kind of the first step in the differentiation process. So we had the stem cell that separated into the myeloid stem line. And when that myeloid stem cell began to differentiate, it became a blast cell. In normal differentiation, the blast cell of a myeloid stem line could have gone on to become a red blood cell or a platelet or a granulocyte, that's the white blood cells that have granules within them, or even monocytes. But because this cancer stops the differentiation, what you end up with is many, many, many of the blast cells circulating in the blood. So the blast cells are what I was referring to when I said baby cells. They haven't grown up yet. They haven't become their full adult potential. So they're baby cells. They are much larger than a typical adult leukocyte. So the arrows in this figure are pointing to the myeloid blasts, the baby cells that are being produced at a very high rate and never given a chance to grow up. Now, myeloid leukemia can either be acute or chronic, and these are very different diseases. Acute myeloid leukemia comes on very rapidly, and it causes much higher rates of cellular division. So clinically, a patient can go from asymptomatic, kind of not clinically having a disease, to within weeks presenting with a full-blown acute myeloid leukemia. It is, of the leukemias, this acute myeloid leukemia is rapidly fatal. If there is no treatment started, patients can die within 6 months of this disease presenting itself. And because it comes on so suddenly and causes so much cellular proliferation, the symptoms tend to be much more severe in acute myeloid leukemia. In chronic myeloid leukemia, it still is uncontrolled proliferation, but the cells have more time to mature. So the circulating blasts are smaller than what you'd see in acute myeloid leukemia. They look a little more like native cells. There's just way too many of them. So the diagnosis is made by the sheer number. There's just way too many of them. So because it comes on slower, the cells are a little bit more mature, this disease can remain asymptomatic for a long time and have really mild really mild symptoms as the disease progresses until it kind of gets to a crossing point, and then you start to get symptoms that get worse. So within each one of these categories, we're talking about very different clinically presenting diseases. So that is the clinical story when we go down the myeloid stem cell line. Now, if we're talking about the lymphoid stem cell line, these are the type of cells that ultimately grow up to become lymphocytes. Now, once we're in the lymphoid cell line, now we're talking about diseases that could either be leukemias or lymphomas. And I just wrote in your notes, the origin of the cancer cells dictates the type of abnormal lymphocyte disease you have, lymphoma versus leukemia. So like where it starts. And this can get extremely complicated, so I'll try to kind of keep it on the surface and not go too deep into it. But the kind of where it starts is how it gets its name, lymph node versus bone marrow. So within the lymphoid neoplasms, we're going to have both lymphomas and leukemias. But let's first talking about the leukemias. Let's get all of these kind of on the same page in your notes, and then we'll go back and we'll talk about the lymphomas. So lymphoid leukemia tells you that it is lymphoid progenitor cells that this leukemia originated. And the leukemia side of it tells you that we are talking about a malignant proliferation of leukocytes. Okay, so the cell line and the type of malignant cell growth, the leukocytes. Lymphoblastic leukemia or lymphoid leukemia is kind of a hallmark, is having lymphoblasts in the circulation. These are similar to the myeloid blasts that we saw in myeloid leukemia, right? They're again baby cells that are getting released into circulation before they've had a chance to grow up. It doesn't matter a ton for us, but for your information, lymphoblasts look different than myeloid blasts. They are typically kind of more solid and they are darker. They're almost all nucleus. When you look at the cell, there's almost no cytoplasm. You will never obviously be making that diagnosis, but if it's helpful for you to know that they can actually look at them and they look differently. So in your notes, the number 2 in your notes says general features of leukemia. So I started with how these diseases are different from the cellular side of things. But once you have the disease of leukemia, they really do share the clinical presentation. So the symptoms are both abnormal growth of white blood cells. So let's talk about all the leukemias we've talked about so far and how this affects the function. So because we're talking about leukemias, we're talking about the cancer, and the problem is in the bone marrow. Now, the bone marrow is where we develop our blood cells. So when you have a cancer that is causing abnormal proliferation of a certain cell type, it's going to be running kind of all the cell-making factories at once and making a ton of cells. And there is a finite amount of space within your bone marrow for all these processes to be occurring. So because you're making so many abnormal cells, they kind of take over the space. And the bone marrow becomes overcrowded with abnormal cells. So the big problem with this is that when the bone marrow becomes overcrowded, it is going to disrupt the function, the normal function of bone marrow. So because it's making so many abnormal cells, we're now going to make the bone marrow worse at making the other cells that it's responsible for. So for both myeloid or lymphoid leukemia, the bone marrow starts making too much of its abnormal cell. We already mentioned that this high proliferation of these baby cells, the blasts, spill into the circulation. So now you have a bunch of extra cells entering the circulation that don't really have the function of what the adult cell would have had. And then kind of letter C in your notes talks about the fact that when the bone marrow becomes so overcrowded with abnormal cells, it is going to impair the bone marrow's ability to make other blood cells. It will impair the formation of cells that you need. Okay, so you have too much of a cell that is really not functional, and then that causes an inability to make enough of the other cells that you need. So with that in mind, it can make sense when you see the symptoms of leukemia. Bleeding can be an issue because they often have platelet dysfunction. The bone marrows aren't making enough platelets. They will be unable to make that important and initial platelet plug. Many patients with leukemia will suffer from anemia. If the bone marrow is overcrowded with abnormal cell making, it doesn't have the space and the resources to make enough red blood cells to keep up. So these patients have reduced red blood cell production. Infection becomes very common because they're making a ton of leukocytes, but they're not functional. So the kind of abnormal, non-functional circulating leukocytes don't have the same ability to fight invaders as the fully differentiated adult cells would have done. So they're more prone to developing infection and being unable to fight off infection. The next three on the list, fatigue, malaise, and unexplained weight loss. I just wanted to highlight those because these three are kind of really big general cancer red flags. These are kind of go into the category of if you see these things, it should be setting off some alarm bells. There's lots of reasons that different cancers cause fatigue. A lot of it has to do with the high metabolism of the cancer, the impact the cancer might have on other functions of other organs. That gets really specific cancer by cancer. In this case, you can see that fatigue could easily be coming from the high metabolism necessary to be pumping out all these extra cells, and then the decreased ability to carry oxygen in the blood from the anemia can all lead to the fatigue and the malaise. If you're unfamiliar with the term malaise, this is a word that kind of loosely translates to like that general feeling of being unwell. I think you've all experienced this. You know that feeling like the day before you get the cold and you're like, something's not right. I know I'm getting sick. And your roommate or your mom or your friend is like, why? Like, what's wrong with you? And you're like, I don't know, but I know I'm getting sick. That's malaise. You've experienced malaise. It is just that general feeling of unwell. Things just aren't right and you know it. And that is a common finding and a big red flag for cancer when it's seen with other symptoms. Obviously, you can get malaise before a common cold and other less threatening issues. In the weight loss, please note the term unexplained. That's the really big part. If there's anything in your life that might explain the weight loss, obviously it wouldn't rise to a symptom. This is the inability to keep weight on, even though you might be eating an appropriate amount, but you continue to lose weight. We'll talk about this right at the end, but the main reason that cancers have so much unexplained weight loss is all of the caloric usage of the cancer. All this metabolism is taking up a ton of energy, kind of sucking that out of the host, and they can't maintain it, and they start losing weight, and the cancer starts stealing from the resources of the body in order to keep the cancer alive. For leukemias, this last one is specific to leukemias. Abdominal fullness is probably the way a patient would describe it. What they're probably feeling is an enlarged spleen. Your spleen is a really important place to kind of filter abnormal cells. We have a really good blood supply that goes through our spleen, and it traps abnormal cells. So when you have a leukemia, it becomes just engorged with abnormal blood cells. And the spleens of patients suffering from blood cancers can be very, very large. All right, so let's move on to lymphoma. So we said cells of the lymphoid stem cell line can develop malignancies of either leukemia if the origin of the problem is in the bone marrow, but it's a lymphoma if the cancer originates in the lymphatics. So with lymphomas, you get tumors that produce masses in the lymph nodes. So lymphomas start in the lymph nodes, but can then spread to other places in the body, like the spleen, the liver, bone marrow, and then in really advanced disease onto even other organs. So this is where I mentioned it can get a little confusing because you can have abnormalities in the bone marrow with lymphoma, and it comes down to where it starts. So when we talk about lymphomas, they really fall into two really large categories. One's large and one is not that large. We either talking about Hodgkin or non-Hodgkin lymphoma. And Hodgkin lymphoma is this is one of the most treatable cancers that you can get, and it is typically, I mean, there's factors about it that make it unique, but its primary marker is that the cancer causes alterations in your B cells. And as it says in the notes, transformation and then loss of typical B cell markers. But the really notable thing about Hodgkin lymphomas is the presence of what's called the Reed-Sternberg cell. This is a really large B cell with more than one nucleus. So you can see the Reed-Sternberg cell right here with in this case, the two nuclei. What's unique about Hodgkin lymphoma is that it progresses in a very orderly fashion. It starts in one lymph node, and then it moves to the next, and then it moves to the next in an orderly fashion. Non-Hodgkin lymphoma is what I was referring to as a very broad category. Under the name non-Hodgkin lymphoma, we're really talking about more than 60 distinct types of cancer. So this is kind of a huge umbrella term, but we call it non-Hodgkin lymphoma when there is a malignant transformation of B or T cells. So unlike Hodgkin lymphoma, the non-Hodgkin lymphomas are typically found in multiple peripheral lymph nodes. So it doesn't just start in one, it's a little bit more widespread, but also spotty. It doesn't move in an order. It's a non-contiguous spread. So it can be in a upper extremity lymph node, and then it's down in the groin. It doesn't have to go in order. It seemingly jumps around. I mean, it stays in one, but then it adds additional lymph nodes as it goes. Of these two diseases, the non-Hodgkin's lymphomas are much more likely to infiltrate other organs. So they get extranodal involvement, which makes them a much more complicated cancer. So lymphomas originate in the lymph nodes, and then Hodgkin's lymphoma is a sort of its own animal where it's just the B cells that change and develop into Reed-Sternberg cells, and all the other lymphomas are referred to as non-Hodgkin lymphoma, and that can be B or T cells. But all the lymphomas originate in lymph nodes. The last one to talk about for the blood cancers is multiple myeloma. And myeloma is a cancer of plasma cells. If you remember, the term plasma cell is a B cell once it starts producing antibodies. I try to give you the memory of saying B cells want to be somebody else. They want to be plasma cells, and as soon as they are, they're producing antibodies. These are the cells that we're talking about that are involved in multiple myeloma. Multiple myeloma originates in the bone marrow, and like leukemias, it also impairs production of other blood cells. So there's a lot of carryover there. So I think this is a really important slide to appreciate the growth of cancer, but also the issues related to treating cancer. So the kind of working hypothesis on carcinogenesis is that the entire population of cancer cells, all of them in that final tumor, originated from a single cell that had a mutation. So in this figure, we have a normal cell. And in this case, we're adding a mutagen here, a carcinogen-induced change. And now we have a genetic mutation. And the genetic mutation has changed the normal cell into a cancer cell. In the very beginning of this lecture, we went through potential genes that might have been mutated. The proto-oncogene, the tumor suppressor gene, genes related to the signaling of apoptosis. Once that alteration has happened and the cell has the mutation, and because many of those mutations specifically impair the cancer cell from undergoing apoptosis, and it usually gives a signal to start dividing like crazy, and the go signal is on and the stop signal is off. Once you've made that change, the cell is going to begin to replicate itself. So this theory is kind of referred to as the evolution of cancer. How does it grow and develop? So if you look at the very first change, we see the tumor cells divide, and now we have two of the exact same tumor cell replicated. And this next tumor cell replicates, and we get two more of the identical tumor cell. Now I'm kind of emphasizing that you get this identical tumor cell because I'm hoping that you remember that when a cell undergoes cellular division for replication, it makes a copy of itself, right? You kind of open up your DNA, so you get the two sides, and you use it to make an exact replica of the DNA of the original cell. So the tumor cell, with its mutation, gets to be exactly replicated in its baby cells. But here is the big problem with cancer. Because the cells are replicating very, very quickly, at a very fast rate, they are more likely to make mistakes in that process. So kind of this arm of the diagram on the lower right, the bottom of the screen, I guess, the reds, are showing a whole line of tumor cells that exactly replicate itself. And the baby, 6 generations later, has the exact DNA of the original tumor cell. But if you follow the path up to the top, you can see that the replication starts making mistakes. There's a new mutation that gets made. And instead of being exact replica, now there's a new mutation. And maybe that mutation gets passed along to many, many baby cells. Or perhaps at one of those additional replications, another new mutation is added. So the colors on this figure are meant to represent a cell that has been altered from its parent. So as the cells replicate and divide and you get more and more cancer cells, you ultimately end up with a tumor. That big collection of cells that are growing together in this uncontrolled, unregulated growth. But if you actually look at the cells within that tumor, they have a variety of genetic makeup, because the rapidity of the cell division and often the types of mutations that lead to cancer make it more likely that during cell division they make new mistakes. So if you go to kind of what's typed in your notes, the point number 2 is talking about the fact that the damage led to a cellular transformation. That was kind of our arrow here, our first change. But then all of these errors can accumulate, that the tumor has it's dysregulation of its cellular machinery is the way I put it in your notes. And what that means is, if we go through what we typically see in the type of cells you can find a tumor, it really lays the groundwork for why cancers are so good at growing and so good at spreading. So first, I mentioned a couple times this uncontrolled replication. So the facilitation of an oncogene or the inhibition of a tumor suppressor gene gives it just a go signal all the time. Divide, divide, divide. So you get a rapid increase in the number of cells. Point number 2 is kind of illustrated in this figure. Because they're dividing so rapidly, they're more likely to make mistakes, and you get a loss of genomic integrity. What that means is that the cancer cells multiple generations later are even further changed from the original parent cell. With the dysregulation of cellular machinery, a common effect in a cancer cell is the loss of adherence with surrounding cells. So our tissues have a certain structure because of the way that our cells are connected to each other and organized. When you lose that adherence, that's what I was sort of referring to in the liver slide when I said the malignant tumor doesn't have as well of a defined border, and it's kind of reaching out and infiltrating into the liver, or the organ in that case. The loss of adherence makes it easier for cells to move. So this really facilitates cellular migration and ultimately metastasis, which is when cells move from one area of the body to the other. This multiple errors on replication ultimately leads to disruption in their well, as it says in your notes, both their biochemical and physical communication with the organism at large. That's the human body. So, but what that really means is you're losing all regulation of these cells. It's not responding to any of the normal signals from, hormones, from the nervous system, from the local environment of that tissue. It becomes separate from the organism at large, so we cannot control it. It's not going to listen to any of our normal signals. So because of this rapid change with each cellular division, the tumors, as they develop, become less and less differentiated. Another way of putting that, if it might be more clear in your notes, tumors become more anaplastic. That's when that cell lacks any appearance at all of its original parent cell. They also typically become more aggressive as they acquire more mutations. So if you look, back at that point 1, 2, 3, 4 above of what mutations are likely to do to cancer cells, the more you get of these, obviously the more aggressive it's going to be. We talked about the loss of caretaker cells and the go signal. So if you've got the go signal and you've mutated your caretaker cells, nothing's catching the mistakes, nothing's fixing the mistakes, and they're just replicating faster. So you can see when you go through those original 4 categories of genes that are typically affected, how they quickly compound each other as you accumulate mutations. All right, go to the next page in your notes. And I just want to relate this theory of the evolution of cancer back to some of the common treatment hardships, I guess I would say, that we have with cancer. So a very common clinical story with cancer is that the cancer is discovered and they try a chemotherapy agent. And the chemotherapy agent reduces the tumor, like the tumor responds. But let's imagine that in this picture, the chemotherapy only effectively killed the blue, I should use a different color. The chemotherapy only effectively killed the blue cells. So the tumor would get smaller, right? Because all of the blue cells in here would die. But perhaps the pink and the purple cells aren't responding to that chemo at all. So if one of the purple cells had metastasized off and had started growing in, let's say, a bone, the chemo might have worked really well on the primary tumor and then have just no effect on the metastasis. And that it can be because the chemo was effective at a certain type of cell in the tumor, but the metastasis can be very, very different from the original tumor, because it can be as simple as, one cell getting to a new place and developing, kind of starting back from the beginning. So this theory kind of helps explain that issue with treating metastasis. It also helps to explain why it's so much easier to treat a cancer when it's caught earlier. The earlier you can catch a tumor, the less time it's had to accumulate more and more and more mutations. Or saying that another way, as it's written in your notes, the longer this cancer has a chance to grow, the more mutations it can get, and it can be more and more resistant to therapy, because you can get new mutations that don't respond to the chemo at all and can become very hard to treat. The last point is talking about recurrent tumors. So what many times happens in cancer is a patient has cancer, they get treated with chemotherapy. It effectively, shrinks the cancer to the point where people are put into remission and the cancer is quote unquote gone. And then a tumor comes back. When they go to treat the returning tumor, it doesn't respond at all to the chemo. Like, it just doesn't have an effect. And if you think kind of this theory through, if the chemo killed off all of the cells that respond to that chemo, then it only left behind the cells that didn't. So when those cells started to replicate again and develop a secondary tumor, a recurrent tumor, it would be made of all cells that are resistant to the chemotherapy. So it's incredibly heartbreaking, because a treatment worked and it got rid of the cancer, and now your cancer is quote unquote back and the drug doesn't do anything. And that's because the drug already killed all the cells that it would kill, and what remains doesn't respond to that chemo. So this figure really just lists out the cancer hallmarks. These are important to know and appreciate. I think they have been fit into our story, so I hope you don't feel like this is a list you just have to memorize. But things that make cancer cancer is that the cells are not killed off by our immune cells. They avoid immune destruction. They don't listen to signals from the tissue that they're near, so they don't respond to growth suppressors. So if you're getting the signal, it's like, nah, I don't care, I'm gonna keep growing. It has the ability to replicate forever. And this is a very unique thing. That's what I mean by replicative immortality. This is very unique. Our cells, our normal non-cancerous cells, don't have this. With every single replication, at the end of our DNA, we have telomeres, and the telomere gets shorter with every replication. And when the telomere gets to a critical length, you will not replicate that cell again. It's how we protect ourselves from having too old of cells in, I guess old is okay. they haven't divided so many times they can make mistakes. Cancer avoids this. They manage to keep their telomeres long, and the cells can replenish themselves as many times as they want. Tumors frequently promote inflammation. This gives a lot of the symptoms of cancer. I'm looking at your list. It kind of jumps around. All right, the next one in your list is the genetic instability. It's much more likely to get errors as it replicates. We mentioned this right at the beginning, but cancers typically induce a ton of angiogenesis because they need a ton of blood flow to support all this cell growth. They're very resistant to cell death. They block their apoptotic signals. They don't respond to external signals telling them to die. They have deregulation of their cellular energetics. This is a fancy way of saying they come up with weird ways of making ATP. That's my physiologist explanation. Cancer cells, typically, a lot of cancer cells develop an aerobic glycolysis. So it's very weird that they can use oxygen and run glycolytic pathways. So that's what it means by deregulating cellular energetics. They get good at making ATP any way they can. They keep their proliferative signals on all the time. And then lastly, I remember why I put it out of order now. Lastly, they can invade and metastasize. And the reason I put it last is because I have a little section just talking about the ability of cancer cells to evade and metastasize. And this truly makes a malignant cell malignant, because a benign tumor does not have the ability to move to a different location. That is very unique to malignancies. So as it says in your notes and on the slide, malignant tumors can spread from their primary site to other locations in the body. The thing to know about spreading cancers is the more rapid the tumor growth, like the faster it's replicating, like if a tumor is growing super fast, it's much more likely to metastasize to other areas. The reason for that is that as the cells are dividing and growing, we shed millions of cells into the cancer sheds millions of cells into the blood supply every day. Okay, so they're getting into the blood supply like crazy. But cells aren't meant to live in blood, right? They about 99% of cancer cells that get into the blood are killed just by the turbulence of the blood flow within 24 hours. But if millions of cells are getting into the blood and 99% are killed, even if 1% remain, that's cancer cells that are living and taking the blood vessels as a superhighway to another place. So the reason rapidly growing tumors are more likely to metastasize is simply the increased number of cells. The more cells, the increase the probability of cell survival to form a metastasis. So cancer can spread in multiple ways that are illustrated on this diagram. The first is that they can seed or penetrate into a neighboring body cavity, so they can kind of move through. So this is a picture, all of the kind of white granules that you see in this are tumor nodules that have moved into the mesentery. So this is a common spreading pattern for ovarian cancer, that it starts in the ovary and then it can move into the mesentery. The next way that it can go, the green tube in our figure, is lymphatics. So cells can get into the lymphatics and then travel that way. When you have a lymphatic vessel, they kind of join up at lymph nodes. So we will often see cancer cells in neighboring lymph nodes, and that's a common test. When you're first getting your cancer diagnosis, they will look at neighboring lymph nodes to see if the cancer has spread. And then the last is in the blood. Oops. So I just listed the 5 most common sites. And one thing to note is, I mentioned that cancers shed millions of cells into the blood supply, and most of them are killed. So if you had to think about it, do you think it's more likely that the cells would die if they fell into an artery or if they fell into a vein? Artery has a much higher pressure and flow, so they're more likely to live in veins, and that you can kind of see some of that in the common sites. So the first is lymph nodes, and that one hopefully is intuitive since cells can travel in the lymphatics. So a lymph node is the next stop down the track, and that's why they take up residence there and can grow. The liver is a really common place for metastasis because of the venous blood that it gets. So all of your GI system, your stomach, your intestines, your pancreas, they all drain their venous blood. The first stop for that blood is the liver, right, via the portal vein. So the liver filters all of the venous blood from our GI system, which physiologically is very good. But because of that, the liver is exposed. If the cancer originated anywhere in the GI system, can be easily exposed to cancer cells that were less likely to die in venous blood than they were in arterial blood. So because the liver has a direct source of cells from other organs via a venous route, that makes it much more likely to develop metastasis. The next one on the list is a similar story, and that's the lung. Most of the venous blood returned to the heart. The first capillary bed that blood will see is in the lung, right? So all of your periphery drains into veins, and all those veins go back into the heart, big, huge chamber of the heart that then pumps it, the next organ it sees. And the reason I mentioned the capillary is because that's where it's more likely to leave the cancer cells are more likely to leave the vessel, right? Because that's the place where you have single cell layer of endothelial cells. And if they're going to sneak out, it's going to be in a capillary bed. So because the lung is the first organ that most of the venous blood in our body sees, it is a very common place for metastasis. Bone is on this list, and it doesn't kind of have as nice of like a story to help you remember it, but the structure of our bone is an incredibly fertile ground for growth. It's an easy place for cells to live and grow, and that's kind of why that makes the list. Brain is the last one, and then the brain is an unknown. The lung and breast cancers most commonly metastasize to the brain, and we really don't yet know why. But those are two of the more common cancers, and brain is the place that they metastasize to, which is why it gets on the top 5. So you have probably heard about staging and grading of cancer. This is a process by which at the time of diagnosis and then at other later points when you re-diagnose, you can try to describe how involved the cancer is. And it says in your notes, the extent of the disease. So the staging process gives information on how fast the tumor is growing, the extent of the neoplasm, and also hints at the prognosis, or I should say allows oncologists to make a pretty good prognosis for a patient. So the TNM classification system is in this chart. Please do not memorize the chart by any stretch of the imagination, but it's nice to know how these are scored. So the T is a score based on the size and extent of invasion of the tumor, from not, leaving at all the way down to very large and invasive and spread to other organs. The N is talking about nodes. So the score indicates, lymph node involvement, either none, nearby, and then ultimately to distant. And then metastasis is just whether or not a metastasis is present. So there's just some kind of general facts. Please do not memorize these, but it's Sometimes I think it's helpful to know the context and what you're more likely to see. So of all the cancers, the most commonly diagnosed is lung, breast, and colorectal. The most prevalent cancer is breast cancer, and the most deadly worldwide is lung cancer. About 1 in 3 people will be diagnosed with invasive cancer in your lifetime. So obviously each of us will be, if you haven't already, will be touched by this in some way, and you will definitely see patients at different ends of the spectrum of cancer. At this point, it's the second leading cause of death in the US. So the cause of cancer is extremely varied. We can kind of put the causative agents into 2 general categories. Endogenous would be genetic. Exogenous are the things that come from our environment or any external source. In general, most cancers aren't just one or the other. It's some combination of environmental, viral, and genetic. So the carcinogens are, agents that are involved in causing cancer, and this is listed out in your notes for you, so I don't need to read everything, but there's a lot of evidence to show the role of viruses as the inducing agent, a number of chemical agents if you're exposed to them, if you're breathing them in on a regular basis. This puts a lot of our industrial workers at risk. Physical agents, obviously the most common physical agent that causes cancer is the ultraviolet rays from the sun. There's a whole host of drugs. I think there's just everything about cancer is just sort of like a punch in the gut and then a second punch in the gut. But the most common drugs that cause cancer are actually chemotherapy agents. So many of the side effects of drugs that treat cancer actually cause cancer. But at the time that's your option, it's worth the risk, but there's very rarely great options. It's just weighing risks and benefits once cancer is in your life. There are hormones. Estrogen is a good example of one that typically is involved in too much cell growth. Alcohol is on here. It generally is related to the orifices that interact with the alcohol, but also indirectly, and we'll talk about this in the liver lecture, but alcohol abuse can lead to liver cirrhosis, and liver cirrhosis can lead to liver cancer. So that's a little bit more indirect, but it's definitely tied in and very commonly seen. All right, so cancer risk factor. The most significant is age. So as we get older, we are less good at replicating our cells without making mistakes. We're more likely to make mistakes upon cell replication, which means you increase the chance of a mutation landing on one of those key genes that lead to cancers. Right now, it is still established that above 50 is considered a risk factor, but some experts are arguing that once you're above 40, that's not good news for me, not to reveal too much about my age, but that is no bueno. The theories behind, I kind of put my top theory first. I shouldn't have done that. The theories behind why aging is more likely to lead to cancer. One of them is just the length of time that you're exposed to a carcinogen. So, you guys have been in the sun every day of your life, but by the time you're 80, right, that's like 60 more years of exposure to a physical agent. So just that cumulative effect of the years of exposure. Another one is the length of time that your body has had to deal with free radicals. So free radicals damage anything they bump into. So if they bump into damage, they can lead to DNA damage. It's kind of the same thing that it just increases the likelihood the longer you've had to withstand a stress that you're likely to have an error. As we age, we do get increases in pro-inflammatory factors that can lead to cancer, and then those age-associated declines in DNA repair, which I already mentioned. So knowing the risk factors is really helpful because we can talk to our patients about this. I will bring this up many, many times, but this was one of my most empowering parts of clinical practice, was that you spend so much time with patients, and in our current medical setting, patients don't spend that much time with any provider. doctors, PAs, they're in and out. They are really running a rough productivity schedule. We see patients 2-3 times a week, 30, 45 minutes at a time. You end up talking, and I just never ever missed a chance to bring up smoking with every single patient that came in smelling of smoke. Tobacco accounts for 30% of cancer deaths. 30% of cancer deaths are from tobacco, and 90% of all lung cancers are because of tobacco. So when I talk to my patients about smoking, I have many things. I often, because they're in PT, would relate smoking to impaired wound healing and chronic pain, because I think a lot of smokers know that cancer is a risk and they're less scared of it. I'm getting on my soapbox. You guys, I can't help it. I don't know if you've heard about this. I have a smoking soapbox, and I just stepped on it by accident. Let me say a few more things and then I'll step off. I feel like a lot of smokers know that cancer is a risk, but it feels like a faraway risk. But if a human being is in front of you and they are in chronic back pain, and you have a chance to explain to them how their smoking might be making this worse and impairing their ability to move forward, impairing their ability to have a successful back surgery, if that's kind of likely where they're headed, that can be a really powerful moment in their life, because this is a here and now issue that their smoking is causing. And now I'm going to step off my soapbox. This will happen many times this semester. I have a big smoking bias. I want it eradicated from our planet, and I'm starting one PT lecture at a time. That's what I'm going to do. I often tell this story. When I was in PT school, both of my sisters were smokers, and I decided to do a campaign, and I sent them an email every single day with a unique and novel fact about why smoking was bad. And I wish I'd saved them. This was like a million years ago. I was probably doing like dial-up to get my email to work. You guys might not know what that is. But anyway, I have been an anti-smoking advocate for as long as I have known that I care about healthcare. So stay tuned. You'll hear more from me. But anyway, it's a risk factor for cancer. Okay, back on track. Alcohol, we already talked about. Obesity makes the list because of the association between obesity and chronic inflammation. And inflammation has the chance of stimulating an environment that's more pro-cancer. Inactivity is interesting because it's kind of the same. So when you live an active lifestyle, activity and exercise is anti-inflammatory. So when you are inactive, you're more likely to have a pro-inflammatory phenotype. Diet nutrition is on here for many reasons, and this could be an entire class, but my quick version is that a poor diet blunts the immune system's natural defenses against genetic damage. So our immune system is less effective at handling genetic damage if it's not well fed. Unsafe sex practices, the data would suggest that the earlier someone starts participating in sexual activity and the number of partners that they have over their life would increases their risk. So it's important to practice safe sex patterns, living in poor air quality. Indoor smoke from household fuels is on here. This doesn't happen that much in our country, but in other countries where they're burning things within the home for fuel, that's a problem. And then contaminated injections. Poverty as a risk factor is just one of those that sort of eats away at you if you really start to think about it. So you are more likely to develop cancer, and you're more likely to die from cancer if you live in poverty. Some of this is related to insurance. If you don't have insurance and you're only going to go to the doctor when you're sick, and that means that individuals who are less supported in their healthcare setting are much less likely to do any kind of screening. So if there's no early screening, that means that by the time they're getting cancer diagnosed, it will be much later. And as I've mentioned, the later it's discovered, the harder it is to treat. So simply because there's less access to healthcare, things aren't being caught that can be caught if we would just support every member of our country and give everybody health care. There's also higher incidences of smoking and poor diet in households that are under the poverty threshold or in the poverty threshold. This part I just can't not put on here, but studies have shown that if we had equal treatment for all people, we actually have equal outcomes. So if everyone was given the same access to healthcare, we would all have the same outcomes. And I could take this slide out of the PowerPoint. This is a preventable issue in our country that I hope we will see change in my lifetime. All right, so cancer signs and symptoms. Early on, many cancers are asymptomatic. They typically the symptoms of any individual cancer will be specific to the organ that's affected. So we'll kind of hit on some of this when we actually talk about specific pathophys of the organs. But generically, cancer has a set of red flags, and hopefully a lot of these should make sense once you get above the age of 50. And like I said, some experts are arguing to say 40, but 50 is still the textbook cutoff. Unexplained weight loss and fatigue, we mentioned those, that constant feeling of being unwell, malaise. Pain at night is a cancer red flag because most pain of musculoskeletal origin should get better at night. Like you're in a rested position, you're well supported. So if pain is getting worse at night or if it's waking you, that's just kind of uncommon for musculoskeletal pain. The presence of a lump or a mass, and then pain that cannot be reproduced. So we have to care a lot about red flags of cancer and risk factors for cancer because we will be treating patients that might be seeing a PT for a pain that's actually cancer, and they haven't yet been diagnosed and they're trying to treat the pain and they haven't yet found the origin. So you will talk about this in ortho, but if you can't recreate the pain during your initial evaluation, it's very unlikely that it's musculoskeletal in origin. Musculoskeletal pain can be reproduced if we push on it, if we stretch it, or if we contract it. If we do all those things on the area that's painful and we can't reproduce the pain, it's probably not a musculoskeletal structure, and that may be, in the right circumstances, a reason for referral. So I kind of just glossed over the unexplained weight loss because we've mentioned it before, but also because the last page of your notes talks about the term cachexia. Cachexia is the loss of body mass that cannot be reversed nutritionally. What that means is unexplained weight loss is you're losing weight, and even if you eat more, you continue to lose weight. So it is very different from starvation. So in starvation, someone is not getting enough food. The lack of food intake will lower your basal metabolic rate, and that will ultimately lead to the loss of fat and lean muscle. Your body will steal from fat and skeletal muscle for energy. But in cancer cachexia, typically the food intake is normal or maybe slightly reduced, but the basal metabolic rate is increased. That is because the cancer has a very high metabolism. So you have a bunch of cellular eating, right, gobbling up the nutrients, and your body can't keep up. For the same way, you end up losing fat and lean muscle. You steal from fat and protein to feed the need. And if you're losing muscle, because skeletal muscle has become a source of nutrition for cells in your body, it can lead to profound weakness. This can lead to the loss of appetite, anorexia, and it can lead to anemia because of the failure to have enough nutrients to make blood cells. So the look of a person from the outside who has a lot of weight loss might look the same between starvation and cachexia, but the source is very different in that one, it's driven by the basal metabolic rate, and the other is driven by the lack of access to food. Cachexia is not only found in cancer, We will bring it back again when we talk about pulmonary diseases. It's very common. But this is just a list of other diseases that can present with cachexia. So it is a clinical phenomenon when diseases are resulting in increased basal metabolic weight. People can, it's kind of wasting away if you've heard that expression clinically. The more medical term for that is cachexia. All right, we've reached the end of the cancer lecture. I am happy to answer any questions in the office hours this week. Again, they're going to be at 1 o'clock instead of noon. Next week's lecture is all the groups are at your normal time. If you had a syllabus that talked about groups being split up, you just come at your normal time. It is a lecture on ICU lines and monitors. I will have an outline for you printed. I'll post it digitally for those of you who prefer to use laptops and tablets, but I'll also print them off in person, so you don't have to worry about printing anything beforehand. And that will be our final lecture of Unit 1. So then our exam will be on Friday at 10 a.m. on D2L. The exam will be using the Respondus monitor. So if you have questions related to that, or make sure that you kind of understand that that's coming. In the first group, the question was asked, and I remembered after that it wasn't asked in the second, if there would be a remote lecture next week, and the answer is no. So I only get you guys for 4 hours per week. So you'll have your 2-hour lecture on Wednesday, and then my other 2 hours of the week are being used on the exam. So you'll take the exam on Friday, and you won't have lecture again until the next Wednesday. All right, I hope that you guys are all having a great day, and I look forward to